So our next topic is gene amplification. So we have studied the steps in rDNA technology. We have summarized all the steps. One by one we are studying. The first one was isolation of gene of interest. Second one is fragmentation. Third one was separation by gel electrophoresis. And the fourth important step is gene amplification. Gene amplification is done by PCR technique. So what is gene amplification? Means increasing the number of copies. So we have a gene of interest, we have only one copy, we are going to make many copies of it. That is called as gene amplification. It is done by the technique called as PCR, polymerase. Polymerase means it's the name of the enzyme, DNA polymerase. That is enzyme that helps polymerization. So polymerase chain reaction is the full form of it. Now this consists of only three important steps. Very simple thing. So the first step is denaturation. What's the meaning of denaturation? DNA is heated to 94 degrees Celsius, high temperature. That what will happen? That is, we have a fragment of DNA which has to be, so if this is the DNA to be separated, that is to be amplified, we will heat it to 94 degrees Celsius. That time the hydrogen bonds will break and the two strands will get separated as single strands. Now this is called as denaturation. Now the second step is primer annealing. Anneal means hybridize, to join. Okay, so here what we do is we will add oligonucleotides, that is 10 or more our number of nucleotides prepare synthetic primers. DNA polymerization that is when DNA replication time we have seen that to start the process it requires primers. So naturally primers will be there at the cell, but this is an artificial process, so we are adding artificial primers, that's all. So oligonucleotide primers are added to initiate the process. So this is the step where the process is initiated. Now these primers will anneal to the 3' end of each strand. Either 3' 5' angle, either 5' 3' strand. So what will happen? The primers will go and bind to the 3' end. Three end bind. Why? DNA polymerase can always polymerize only in 5' 3' direction. And the DNA strands are having anti-parallel polarity. So this strand should be 5' 3' type. So it will attach to this end. So the primers will attach to the 3' end and initiate the process of polymerization. And this is done at a low temperature. Now the third step is called extension or extension of primers. If we attach a primer, complementary pairing and search, it will extend up to this. Here it will extend up to this. So that is called as extension of primers. So this is actually the DNA polymerization step. So it requires a polymerizing enzyme. Here we use a special polymerized enzyme called as TAC polymerase enzyme. Why we are using TAC polymerase enzyme? Because this polymerase is thermostable, means it can withstand high temperature. This is done at 72 degrees Celsius. This step is done at 72 degrees Celsius. So ordinary DNA polymerase enzyme cannot withstand this high temperature. So this enzyme is used. So why this enzyme, this polymerase is able to withstand high temperature? Because it is extracted from a hot spring bacteria called as Thermus aquaticus. T for Thermus, aquaticus in the aqua. That's why it is called TAC polymerase. So since this bacteria is a hot spring bacteria, the enzymes in the bacteria are thermostable. So we have extracted the polymerase from this bacteria. So that is used. Then magnesium ions are also needed. Then we have to add deoxyribonucleotide phosphate. DNTPs are also added. Now deoxyribonucleotides. Nucleotides are added. 4 different types of nucleotides are added. Then according to the complementary base pairing, it will join to it and finally it forms strands. Double stranded DNA. So one fragment of DNA, one double stranded DNA, this process we start one cycle complete, that is 3 steps complete, we have 2 DNA strand, double stranded DNA, each having one old and one new strand. Now, this this cycle is repeated to 20 to 30 times. So within 2 to 3 hours, we will get billion copies of this DNA of interest. So that is what is done during this PCR by PCR technique. That is how we will gene amplify the gene of interest.